Okay, hi everybody, this is Lilith. I'm one of your eyes for BIPN 160. This is the review video for Lecture 6. Lecture 6 involves a lot of memorization and some research studies, so I will try to cover the main points and put them together in an understandable way. Okay, so let's begin. In Lecture 6, we started to talk about the brainstem. Know that the brainstem contains the thalamus, the midbrain, the pons, and the medulla oblongata, and it is located right at the stem of the brain. Below the medulla is the spinal cord. Let's go ahead and talk about the medulla, pons, medulla oblongata first, and then we will talk about the thalamus. So the medulla has various functions, right? It takes care of respiration, which is breathing, blood pressure, blood acidity. It allows for us to have reflexes in our throat, such as the gag reflex, and it also contains the pyramidal decussation area, which is, I think on the picture, it looks like it's right here. And these pyramidal decussation areas are places on the medulla where the motor neurons coming down from the brain and into the body are able to decussate, okay? The two important functions that Dr. Root described in class were the blood pressure, how medulla takes care of and modulates blood pressure of an individual, and also respiratory control. In short, high blood pressure is sensed by baroreceptors, which are stretch neurons in the carotid bodies of the carotid arteries in our body. These baroreceptors then signal the glossopharyngeal nerve, and then the glossopharyngeal nerve travels this stimulus to different nuclei, and eventually the nucleus of the vagus nerve is activated. And remember that the vagus nerve takes care of a lot of the parasympathetic activities, and because of that, the vagus nerve is able to slow down the heart rate and also dilate arteries. And all of these functions that the vagus nerve can do allow for the lowering of blood pressure. Okay, this basically sums up what Dr. Root wants you to know, and this is exactly what I just said. Baroreceptors are stretch neurons in the carotid bodies. They sense that the blood pressure is high because the walls of the arteries are stretched. There is a stimulus that is relayed along the glossopharyngeal nerve to, I believe this is a type of a nucleus, but I don't remember what it stands for. I don't think you really need to know it, but just know that the information relays through the glossopharyngeal nerve, and it eventually activates the vagus nerve, and the vagus nerve is able to reduce the heart rate of the individual and dilate blood vessels. And by doing all of this, you will decrease the blood pressure. Okay. The second function that is important for you to know about the medulla is respiratory control. Know that medulla is not the only part of the brainstem that takes care of respiratory control. Pons also does this. But the distinction between them is that medulla sets the basal rhythm for breathing, as in the normal way that you breathe when you're sitting at a table. That is what medulla sets, right? Your basal rhythm. And then pons has nuclei that can control or modulate the breathing rate. So if you're starting to run or you have anxiety, then your breathing rate increases, and it is the nuclei in the pons that are responsible for this modulation. Okay, let's think about the Prebotzi complex, which is pretty important for you to know in this class for respiration. So here's a picture of the nuclei in the pons that are more modulatory towards the respiratory rate, and here are the nuclei in the medulla that set the base level of breathing. Okay, but both of their projections allow for us to breathe normally. Okay, The pre-Botzi complex is in the medulla, right? And as I said, it takes care of respiratory rhythm, so it sets the respiratory rhythm. You can think of pre-Botzi complex as an area in the medulla that is needed for somebody to breathe. So if the pre-Botzi complex is inhibited, then the individual or the mouse, if you're doing mice studies, will eventually stop breathing. Okay? I believe Dr. Root mentioned that pre-Botzi complex and this area, we didn't really talk too much about it, but they are both susceptible to many neuromodulatory molecules, and we will talk about the dorsal raphe and locus coeruleus in a little bit in the next page. But from this slide, what you really need to know is that pre-Botzi complex is responsible for us to be able to breathe. It can have a lot of outputs to different areas of our body, and it also receives a lot of neuromodulation. Now let's talk about the studies that looked at respiration that Dr. Root talked about. In this study right here, if you're looking at the slides from lecture, what happened was they used the optogenetic tools that we talked about, halorhodopsin and channel rhodopsin. However, they didn't use those same exact ones. They used something that is called the ARCT, and this is an inhibitory optogenetic tool. So once you shine a light on this optogenetic tool, then they were able to inhibit the pre-Botzi complex of this mouse. And by inhibiting the pre-Botzi complex, you see how this mouse was breathing, and then there is a cessation of breathing. So the breathing stops. And then in this scenario, they used an optogenetic tool that is excitatory. So when they shined light and they excited the pre-Botzi complex, this is the regular mouse breathing, and then you see a drastic increase in breathing because the pre-Botzi complex was stimulated. So this is already proof that pre-Botzi complex is not only involved in breathing, but is also required for breathing. Because if we take it away, the animal stops to breathe. Okay. This study right here talked about two molecules. So fentanyl is a drug that can stop you from breathing. And how does it do it? Fentanyl is able to bind to the pre-Botzi complex, and it inhibits or suppresses the pre-Botzi complex. And as we just said, if you suppress the pre-Botzi complex, then this is normal breathing, and then your breathing stops. However, if there's not enough of this fentanyl given to this person, then breathing comes back. But it's this drop and suppression in breathing that tells us that fentanyl does inhibit pre-Botzi. So that's what A tells us. What does B tell us? B tells us that if fentanyl is given to this mouse, then their breathing should stop at some point, right? So we should see this dip, but we don't. Why? Because right when we get to this point, this other molecule that's called naloxone, I don't know, I think it's blurry, but I think it's spelled naloxone. I think you'll see better on your lecture slides. This naloxone is an antagonist for systemic fentanyl induces respiratory suppression that is blocked by antagonist. Yeah, so naloxone binds to the pre-Botzi complex, and it doesn't let fentanyl to bind to it. So when naloxone is present, then the effects of fentanyl are no longer affecting the pre-Botzi complex. So the breathing never stops. Okay, so essentially main point, naloxone reverses or doesn't allow fentanyl to do what it does, which is suppress the pre-Botzi complex. Okay, and then the last study that we talked about, or part or experiment, research experiment, was how it was found that opioids are actually binding to different neurons in the pre-Botzi complex. And they tried to figure out which neurons the opioid or the opioid agonist, which is a type of a molecule that structurally is similar to opioid. They tried to figure out which neurons does it bind in the pre-Botzi complex. And they figured out that it is this neurokinin 1 receptor containing neuron, because when they tried it on different neurons, they saw that it is only the neuron that is expressing this neurokinin 1 receptor that actually makes the mouse to stop breathing. So that's how you can tell that opioid agonists or opioid itself acts on the neurokinin 1 receptor neurons of pre-Botzi complex. And again, whole point is pre-Botzi is inhibited by this opioid agonist, so breathing stops. Okay. Then the next topic that we talked about is the pons. There was really not much to discuss research-wise for pons, but some things for you to remember. So there are medullary and pontine reticulospinal tracts in the pons, and these are responsible for two functions. They're responsible for pattern-based movement, meaning walking or running, and then it is also responsible for pain modulation. And I think Dr. Root said that opioids also bind to neurons in these tracts because opioids are against pain, right? So pain modulation neurons are also located in these tracts. And then the pons also contains the dorsal periaqueductal gray, I believe. That's what the area is called. And this area responds to threats. And it responds to specifically fast type of threats. So if the mouse is looking at a screen where something just dark and scary pops up, then this area in their pons is activated and it stimulates their escape response. So they want to run away. However, if this area is inhibited, then inhibition blocks the escape response and they no longer run away when they see a threatening image. Okay. Then we went ahead and talked about the 4 major neuromodulatory systems in the brainstem. Some of these are in the midbrain, some of these are in other places, but I think what you mostly need to do is just commit to memory or know exactly where these definitions are in your notes, because this will be easy points on your exam if you're asked about these. So let's go over them. We have serotonin that signals reward, dopamine that signals reward prediction and seeking, motivation and movement control, norepinephrine signals arousal and attention, acetylcholine signals wakefulness, arousal, attention, memory, and motivation. Okay, and this really is just something that you have to commit to memory, or again, because your exam is open notes, just know exactly where this is in your notes. We will come back to these when we start to talk more about the midbrain, but before I get back to these, I want to mention a couple structures in the midbrain. The superior and inferior colliculi in the midbrain are areas for motor eye control, so controlling your eye muscles. That is controlled by the superior colliculus, and then the inferior colliculus is controlling auditory processing. An easy way for me to memorize this is on a human body, the eyes are more superior to the ears, a little, or now that I'm thinking about it, I guess. Well, I always think about it. The eyes are a little more superior to the ears, so the superior colliculus has to do with eye motor control, and the inferior colliculi have to do with auditory processing. You don't really need to know the pathway of the auditory processing just yet, but we will talk about this in the coming lectures. And the second structure that is important in the midbrain is the pineal gland. The pineal gland is important because this is how we know when it is light outside or around us and when is it dark. When the pineal gland is stimulated, because our eyes see darkness around us, so there is no light going into our retina, then the pineal gland is, I think, either stimulated or simply not inhibited. So when this happens, the pineal gland produces this molecule called melatonin, and melatonin allows us to become more groggy and want to sleep. However, when there is light around us and the light photons are entering our eye and hitting the back of our eye, the retina, then there are neurons that are stimulated, and then they send this stimulation through a couple nuclei that we will learn later in coming lectures. And those stimuli, because they were stimulated by light, they end up inhibiting the pineal gland. So the pineal gland does not produce melatonin, and the person does not want to sleep. That's why it is hard for people who just start working night shifts, for example, right? If you go work in a night shift in a hospital, then for the first week, you're going to have a really hard time sleeping at daylight because your body just isn't used to producing melatonin when there is light outside. Okay, so hopefully that helps you remember. Now let's go and talk about these neuromodulatory molecules that exist in our brainstem, and they all come from their own nuclei. So dorsal raphe nuclei produces, and I've color-coded the arrows, so follow the green arrow. Dorsal raphe produces serotonin, and because serotonin signals reward, dorsal raphe also signals reward and pleasure. He didn't mention it here. The VTA, ventral tegmental area, and the substantia nigra are nuclei in the brainstem that are dopaminergic neurons. See right here, follow the blue lines. Dopaminergic, they produce dopamine. And both of these nuclei control signal reward expectation or prediction, like you're predicting or expecting that you're going to get a reward. That is when these dopamines are produced. And substantia nigra also signals initiation of movement. And when neurons in the substantia nigra are destroyed or inhibited or lost, then this causes the disease Parkinson's disease. Okay? Parkinson's disease is the one where the individual is, I believe, has tremors and has a difficult time to initiate or start or stop movements and control movements. Then if we follow the red arrow, norepinephrine is being produced from the nucleus locus coeruleus. Noradrenaline is noradrenaline or norepinephrine. Adrenaline and epinephrine are the same thing, just different names. They signal arousal and attention. And then the basal nucleus secretes acetylcholine. It has cholinergic neurons, and those signal wakefulness and arousal, and the other functions that are mentioned right over here. Okay, so all of this is just something that you have to memorize, or hopefully you have taken good notes for the exam. Then we talk about the thalamus, and we talk about the different types of the nuclei in the thalamus, and each nucleus has their own function. And this is something, again, that you want to memorize, both the functions of the nuclei and also their inputs and their outputs. Okay, I've highlighted the names of the nuclei over here, and I've also highlighted the nuclei over here, so you can see which structure takes input to each nucleus. And then I've highlighted the nuclei over here, so you see where the output of this nucleus goes to. Okay, I won't go over these, because you should probably just read them and try to memorize them, or again, just have them in your notes and know where they are. Me going over them won't really benefit you as much. But I will leave this here. Hopefully my notes help you also. And then another thing that I wanted to mention, you might be asked this on the exam, the internal capsule and the corona radiata. These are both white matter, and they are basically axon projections that travel from and to the cortex, to or from other areas of the brain. So this is mainly how different parts of the brain are connected to one another through the internal capsule and corona radiata. Okay. I think these are the main topics in this lecture. You definitely want to, I know your test is in an hour, and I'm sorry that I posted this so late. It's been a crazy week. But you definitely want to have your notes by your side for the exam. The notes that you took, the lecture notes, have the practice sets, have the practice test, last year's test next to you, and best of luck. That's all I have to say. Don't hesitate to type your questions in the discussion or email the professor or the IAs if you have any questions during the exam. Okay, we'll be there to help. Good luck, and I'll see you for lecture 7.